Good morning, children. Today our topic is thalassemia. Now, this is the first genetic disease which we are doing. By now we know that what are the causes or what is the reason behind a genetic disease. So when there are mutations in the genes of a chromosome, then it leads to genetic diseases. If the mutation is in the autosome, then it's an autosomal genetic disease. If the mutation is in the sex chromosome, then it leads to a sex-linked genetic disease. So what is thalassemia? Let us see that. Thalassemia was first discovered by Dr. Cooley in the year 1925. It is a hemolytic anemia. What is the meaning of hemolytic anemia? Hemolytic means when there is destruction of RBC, and anemia means when there is less RBC. Okay, when there is less RBC or faulty RBC. So less number of RBC or faulty RBC or destruction of RBC has been caused because of faulty synthesis of the hemoglobin. We know what is hemoglobin. Hemoglobin is the protein which makes up the blood, which is found in the RBC. So RBCs gets generally destructed or there is less number of RBC. It is because there is faulty synthesis of hemoglobin. Now, what are the causes of thalassemia? The first thing is that it is a genetic disease which is caused by autosomal recessive gene. So that means it is an autosomal genetic disease and the allele is a recessive allele. Or we can say it is a recessive gene which is actually causing this autosomal disease. Now, what is the number of chromosome? What is the chromosome number in which the defect occurs? So, the defect in the gene occurs in chromosome number 16 and chromosome number 11. Now, why is this defect or why has this defect occurred? Because there has been mutations in the genes present in chromosome 16 and chromosome 11. And what has that mutation caused? That mutation has caused defective synthesis of alpha globin chain or beta globin chain of hemoglobin. So we know that hemoglobin is a protein. So this alpha globin or the beta globin, they are the components or subunits of protein which is forming a part of the bigger protein hemoglobin. Okay, and this hemoglobin is actually made up of 4 polypeptide chains, out of which the 2 polypeptide chains are alpha globin chain and the other 2 polypeptide chains are beta globin chain. So mutations basically occurs in those globin chains. Okay, and thus there is defective synthesis from those globin chains that causes thalassemia. Clear? Thalassemia is of 3 types: alpha, beta and delta. What happens in thalassemia, or how do we understand that a person is having thalassemia? What are the symptoms? Now if you just look at the diagram, you see this is how a normal red blood cell looks like, right? These are white blood cells, these are RBCs. When it is thalassemia, just look at the malformed red blood cells. That means it is completely a faulty RBC. There is destruction of RBC or less number of RBCs produced in case of thalassemia. Right? So what are the symptoms? Synthesis of less hemoglobin or formation of abnormal hemoglobin. Why? Because the mutation occurs in the globin chain of hemoglobin and that can lead to less hemoglobin and that can lead to abnormal hemoglobin. And this less hemoglobin and abnormal hemoglobin will lead to less RBC in the body. Premature destruction of RBC. So the RBC just gets destroyed before time, and so there is development of anemia. Hepatomegaly and splenomegaly, that means there is enlargement of liver and of spleen. There is skeletal deformation and iron overload in the body, because this hemoglobin is nothing but is actually carrying a iron part, the metal. And then there is retardation of the body growth. Clear? Now let us see the inheritance of thalassemia. So let us denote the genotype, or we can say let us take the two alleles as the dominant one as capital R and the recessive one as small r. Right? Now, when I say that thalassemia is an autosomal recessive disease, that means the recessive allele, that is small r, is responsible for it. So, to express the disease, you need to have both or a homozygous recessive condition, that is small r, small r. Clear? And when a person is a carrier, that means it is carrying only one copy of that recessive allele, that is one small r. So here it is an unaffected carrier father. That means the father is a carrier. So he is only carrying one small r. So we can see he is only carrying one small r. And then one is a capital R. Right? So capital R is not responsible for thalassemia. So the person doesn't show any symptoms, but he is just carrying the allele. Right? Similarly here, it's an unaffected carrier mother. The mother is also not showing any kind of symptoms because it is a heterozygous condition carrying only one small r. Now, when there is a cross, and when they are breeding and the offspring is found, first offspring is happening capital R, capital R. So we know that thalassemia can only occur when it is in a homozygous condition. So there is no homozygous recessive allele. This is completely homozygous dominant. So it is unaffected. So this is a normal child. This one is capital R, small r. So we know it is having only one copy of the recessive allele. It's a heterozygous condition. So when it's a heterozygous condition, the offspring is a carrier. Similarly, with the second offspring also, if it's capital R, small r, this is also a heterozygous condition carrying only one small r. So this is also a carrier. But the last one is having the homozygous recessive allele. So this child is actually having the thalassemia because it has both small r, small r. And I told you that thalassemia is an autosomal recessive disease. That means for the disease to express itself phenotypically as well, and the person is not a carrier, the person is having a homozygous recessive allele. That means it's in a homozygous condition, the recessive allele. So it is small r, small r. So there is one which is completely normal, two they are carriers, and one is completely thalassemia. Clear?